Hello, my dear students. I'm Dr. Vaishali Parambe. I've been teaching anatomy for last 25 years and I love it. This lecture is a part of my Histology lecture series, part of epithelium. Okay, so epithelium, this is epithelium part 2. We'll come to what we're going to cover today, but first let's think, what are tissues? Tissue is a collection of cells which are functioning in a collective manner. It's a group of cells which are aggregated and they function in a collective manner. So, human body has got 4 types of tissues: epithelial, connective, muscular and nervous tissue, out of which we are at the epithelial tissue. So, what do you understand by epithelium? Epithelium is a collection of cells, yes. Closely opposed to each other, yes. Forming a coherent layer, sensible layer. Can you have random cells coming together? No. They're all forming a coherent layer, either lining the body from outside or lining the body from inside. Okay, that is epithelium. Based on the number of layers of cells in an epithelium or based on the shape of the cell on the top, you can classify epithelium variably. So, if the layer is single layer of cells, you call it simple epithelium. If you have multiple layers of cells, you call it stratified epithelium. Got it? So, single layer simple, multiple layer stratified. Now, what is the type of cell on the top of epithelium? If it's a flat cell, you call it squamous. If the height of the cell is equal to the width of the cell, you call it cuboidal. If the height is way more than the width, you call it columnar. So, putting these two together, you have following type of epithelia: simple squamous, cuboidal, columnar, pseudostratified epithelium. 1. These are simple epithelia, single layered. And you have stratified squamous, stratified cuboidal, columnar, transitional. These are multi-layered epithelia, which is what we are discussing today. Simple epithelium has been discussed in epithelium lecture part 1, the link for which is given in your description section. I insist that you go and see that lecture as well. Right. So let's begin to discuss stratified epithelium. पहले मुझे क्या आता है? What do I know? I know that it is stratified epithelium. That means it will be multiple layers. Am I looking for a single layer? No. I am looking for multiple layers. If the top layer will be flat, I will call it stratified squamous. If it will be cuboidal or columnar, I will call it stratified cuboidal or stratified columnar. This much I know. Okay? Always go into the topic asking yourself, what do I know? Okay? So, stratified epithelium, some basic features. It is found at places where there is a lot of wear and tear. For example, I will have stratified epithelium on my palm, because my palms are all the time doing work. Okay, so a lot of friction happening. So I need multiple layers. Okay, if I had a single layer, that would be lost because of amount of movement I do. So it's found at places where there is wear and tear. Okay, number of layers is decided by amount of stress. We'll discuss this again later. It's beautiful how our body has made itself in a way that it is all the time functioning for protection of this body. Okay, beautiful. I want you to see the beauty, not just the amount of studies you have to do. Right. So, what is the function of stratified epithelium? Obviously protective. Can it be absorptive? No. How will you absorb through so many layers? Can it be secretory? No. It is basically protective in function. Based on the cells on the top, you can have stratified squamous, stratified cuboidal or columnar, and a third type of epithelium called transitional epithelium. I'll be telling you details about this. So let's begin to study stratified squamous epithelium. Again, मुझे क्या आता है? Stratified है तो multiple layers होंगे. Squamous है तो cell on the top will be flat. Clear to you? Okay, so let's see let's understand this. It's a multi-layered epithelium which is characterized by constant formation and movement of cells Who is forming the cells? Cells are formed in the basal layer, this layer. Okay, this is the layer that is all the columnar cells which are all the time multiplying, giving rise to new cells that will be rising upwards. The cells rise upwards, holding on to each other with the help of desmosomes or cell junctions, and they rise in the form of a continuous cellular sheet. As they rise up, they change shape from columnar to polygonal to flat. Can you see that happening there? Okay. So this layer gives rise to cells. They hold on to each other with the help of desmosomes. They rise up in the form of cellular sheets, columnar to polygonal to squamous. Right. So stratified squamous will therefore be found at places where there is maximum mechanical stress felt. Function is protection against mechanical, microbial or chemical changes. Right. Stratified squamous epithelium can be subdivided into two types, keratinized and non-keratinized. What is this, ma'am? What is this keratin? What is this non-keratin? I'll tell you what it is. Right. So take a look at this image. Can you see that the bottom layer is same as what we saw earlier? There are the same type of cells which are going from columnar to polygonal to flat. But cells here seem to have lost their nucleus. There is no nucleus here. This big fat layer on the top, which has got no nuclei and which is actually dead cells, is called as keratin. And it is the presence of this keratin which decides whether the epithelium is stratified squamous keratinized or non-keratinized. So presence of keratin is stratified squamous keratinized epithelium. Clear? We'll learn some more about it. So, I've taken the same section and put it here for you to understand. Multiple layers seen. The bottom layer is called stratum basale. This is where cells multiply. They begin to rise up, remember, in the form of cellular sheets. These sheets hold on to each other with the help of cell junctions called desmosomes. As they rise up, during the processing of the slide, some shrinkage of cells takes place and only the cells remain attached at the points of desmosomes, because of which the cells seem to show spines, causing it to be called as a stratum spinosum. As they begin to rise up, they begin to pack themselves with filaments called keratin filaments. Okay? As they rise some more, cells collect granules in them. These are keratohyalin granules. Don't have to remember everything, just understand the concept. Cells are gathering granules in them called keratohyalin granules. Because they show granules, the layer is called stratum granulosum. So, stratum basale basal layer, stratum spinosum, stratum granulosum. Okay? As the cells are rising up, columnar, polygonal to flattened cells. Now what happens? The cells above granulosum begin to lose their nucleus. Can you see that there is hardly any nuclei here? Meanwhile, the cell is also releasing a lipidic content all around. Okay? This lipidic content is present extracellularly. Cells are held on to each other, but because of lipid, the layer becomes impermeable to water. So, if you are in water, does your body absorb water? No, because there is a coating to it which is impermeable, or waterproofing of your body is done by this lipid, glycolipid. Right above this now, cells completely lose their nucleus. Okay, they are flat plates of cells holding on to each other with the help of a glycoprotein lipid complex. This layer on the top is called as stratum corneum. Basale, spinosum, granulosum, lucidum is the one which lipid layer, and stratum corneum. How do you summarize this? Strongly attached layers of living cells. On the top, superficial layer of dead plate-like cells held together by glycoprotein and lipid complexes. It's found at places where there is friction but the surface is dry. For example, your skin. This epithelium is therefore called dry epithelium. Okay, I want you to do something. If you've not moisturized your skin, just run your hand on your skin. You will find white lines getting created on it. Of course, I have put a moisturizer, so I can't tell you how, show it to you on my own skin. But if you do that, you will find white lines appearing. Why is that happening? Because the keratin is coming off. This layer of stratum corneum is protective. If you give friction, the keratin sacrifices itself, protecting the underlying living layers. Got it? Now let's understand a little bit more. You know that epithelium of skin is stratified squamous keratinized. But number of keratin layers will be variable. In your palm or in the plantar aspect of your foot, you have a nice thick keratin. Why? Because more friction. You're walking on that foot. You're using your hands. So much friction. You have to have thick layer of keratin. Whereas the layer of keratin on your forearm or on your face is thin. You don't have that much friction. So the thickness of the keratin layer will be variable depending upon amount of friction. One more interesting. Do you know what is a callus formation? Okay. So suppose you have a ill-fitting footwear. Chappal, thoda sa the nail of the chappal is hurting your foot. After some time, a kind of callus gets formed there, a thickening of the skin. Why? Because keratin gets deposited there, trying to protect the underlying epithelium. That is how stratified squamous keratinized epithelium functions. It's beautiful. Try to understand it. Don't just mug it just because it is something you have to study. Right. So revise stratified squamous keratinized epithelium, bottom layer of columnar cells which will multiply. They will give rise to cells which will rise up. The cells are attached to each other with the help of desmosomes. Okay. The little bit of shrinkage of cells cause them to look spiny, hence called stratum spinosum. They have started to pack themselves with keratin filaments. Rising up, cells contain granules in them, keratohyalin granules, hence called stratum granulosum. Next layer is a lucid layer which contains lipid in it, waterproofing your skin. And the last layer is a layer of dead cells, plate-like, no nuclei, thick protective layer. The cells on the top are all the time desquamating off. This is called stratum corneum. So this is your stratified squamous keratinized epithelium. Right. What is non-keratinized then? Obviously no keratin layer. Okay. So same thing. Basal layer, cuboidal polygonal cells, flattened cells on the top. But do they have dead cells? No. Right on the top, you will see nuclei are contained in the squamous cells. There are no cells with no nuclei there. So that is the difference. There is no keratin formation. There is no dead cell layer formation. Okay. They are also shed off just like keratinized epithelium, but they are found at places where the epithelium is moist. Your oral cavity. Roz to khate ho. Every day you eat food. So much friction in your mouth. Okay, but because there is a moisture, there is no keratin layer formation. Okay, so where will you find stratified squamous non-keratinized epithelium? Oral cavity, esophagus, anal canal, vagina, cornea, conjunctiva. All these are places where you get non-keratinized epithelium. Okay. Finally, if you have vitamin A in adequate quantity, it performs the function of maintaining epithelial integrity. Reduction in vitamin A causes your skin to become dry, pruritus, even corneal ulcers. Remember, the government is fortifying your food with vitamin A to prevent this. And this is something interesting, epithelial swab. Sometimes you will see that doctors take up a swab. They take a sample of your skin cells by just running the swab on your skin. This is especially true in case of vagina, where we do something called as pap smear. I'm sure you've heard about it. So what happens is cells are all the time being shed off. Then why go take biopsy? Just run a swab on the skin, you will end up picking up the cells and you can examine them for any cellular changes. This is called exfoliation, and this method is called exfoliative cytology. So students, what have we learnt? We have learnt about stratified squamous epithelium. Initially, we just classified the epithelium and described its basic features. Then we began to talk about keratinized epithelium. If you are writing a note, first draw the epithelium, then discuss each layer: basale, spinosum, granulosum, lucidum, and corneum. Describe the locations where it is found and why it is found. You can discuss thickness of keratin also, like we did today. Then non-keratinized, again locations where it is found and why it is found there. Finally, applied anatomy, vitamin A deficiency, exfoliative cytology. Remember to draw both the diagrams, keratinized as well as non-keratinized. Right. So with this, we finished stratified squamous epithelium. Let's go to the next topic, which is transitional epithelium. Students, if you're liking this lecture, do like and subscribe to my channel. Let's now come to stratified cuboidal or columnar epithelium. Basically, this is found in ducts. Okay, so ducts of sweat glands, salivary glands, mammary glands. You will find 2 to maximum 3 layers of cuboidal to columnar cells. Let's come to an amazing topic, the transitional epithelium. Transitional, ma'am, it's going to transition? Yes, it's going to transition. I'll tell you how it does that. Come on. So it's a very, very common short note. Please prepare it separately for your exams. So this is a stratified epithelium, multi-layered, but it is barely 4 to 6 layers thick, consisting of a basal layer of columnar to cuboidal cells. Cells on the top are polygonal large cells. These cells in the base might show mitosis because this is the layer which multiplies to give rise to higher layers. The next layer is polygonal cells. They are larger than the columnar cells but are smaller than the layers on the top. So what is there on the top? The top layer is superficial layer of large umbrella-shaped cells. Okay, you can see umbrella, that is only to trigger your memory. So these are your umbrella-shaped cells. Some of them might even fuse together to form binucleate cells as seen here. The cells on the top bulge into the lumen in the form of scallops. Okay, scallops. I've shown what is scallops for those of you who don't understand. The top of the cell is coated by a glycocalyx or a cuticle, a layer that is protective for the epithelium. Okay, so this is the glycocalyx or cuticle. Remember to draw this in your exam. Okay, so this layer protects the underlying tissue from the toxic effects of urine. So one function of transitional epithelium, protective. Okay, all the cells on the top are attached to each other with the help of cell junctions. What is that importance of this? It will not allow absorption. So the toxic urine will not be absorbed deeper below. So what function are you seeing? Non-absorptive, protective, non-absorptive. I want to take you to its third important function, stretchability. I think all of us are aware of the ability of bladder to stretch. Bladder is lined by transitional epithelium. So just imagine that you have been drinking a lot of water and some more water, and slowly your bladder goes on expanding. Does your bladder say, I cannot expand? No, it expands till a point where it cannot expand anymore. It has good amount of stretchability. So transitional epithelium has ability to stretch and relax as per requirement of that organ. Here you are seeing the relaxed, unstretched urothelium or transitional epithelium. It's about 4 to 6 layers thick. But when it is stretched, it stretches in such a way that it almost appears like it has fewer layers, maybe 3 layers. Okay? It has immense ability to stretch. So what are the properties or functions of transitional epithelium? Stretchability, non-absorptive, non-secretory, and protective. These are functions of transitional epithelium. Transitional epithelium is called transitional because it is said to be somewhere between stratified cuboidal to columnar. More than that, it can transition from relaxed to stretched to relaxed easily. Hence, it's called transitional epithelium. Is there another name for it? Yes, another name is urothelium. Obviously, because it's an epithelium that lines your urinary tract. Question: exactly where is it lining your kidney? No. It is lining the major and minor calyces, the pelvis of ureter, the entire ureter, the urinary bladder, and the urethra partly. These are the locations at which transitional epithelium is found. Please learn this up. There is no forgiving for not knowing these basics. Transitional epithelium does have ability to regenerate, but it's a slow regeneration. Remember, this is an epithelium that tends to undergo malignancy in the form of papillary tumors. This is associated with aniline dye industry, typically found in this industry or exposure to benzidine. So with this, we come to the end of transitional epithelium. Students, this slide gives you an entire list of points on which you should discuss this. This is a very important short note. Please take a screenshot of this and revise. Okay. So first, classify the epithelium. Describe all the layers: basal, intermediate, superficial. In superficial layer, talk about umbrella-shaped cells, binucleate cells, scalloping, the glycolipid, then discuss the properties of the transitional epithelium, discuss why is it called transitional, where is transitional epithelium found, another name for transitional epithelium, and so on. Use this slide well to give you a full marks in transitional epithelium short note. What are the questions asked on this topic? Short note on stratified squamous epithelium, short note on transitional epithelium. Of course, identify epithelium will come in practical routinely. Each type of epithelium I have told you can be asked. Related lectures that you should be watching. One is a lecture on glands. Glands is epithelium 3 lecture. You must watch it. Epithelium part 1, where we talk about simple squamous, columnar, cuboidal, and pseudostratified epithelium, are also a part of the lecture group under the topic of epithelium. The link for these lectures is given in the description section. Do watch it. Students, if you like the lecture, do like and subscribe to my channel. Share your thoughts in the comment section. I really want to know if my lectures are helping you in your study of anatomy. So thank you very much, students. See you across the screen in some other video. Bye.